Hi, my name is Madison Sheets, and this is my journal club, and I have 5 people here that are going to listen. So if y'all could introduce yourselves.
I'm Amy Smith. I'm Morgan McCoy.
John Goings.
I'm Grace Wynn. And Ashlyn Balthazar.
Okay, so here we go. The title of my journal article is a Phase 3 randomized trial of Velexator in sickle cell disease, hemoglobin oxygen affinity modulation to inhibit HBS polymerization. All of that is summed up in the word HOPE. That's kind of what they've deemed this trial. It is published in the New England Journal of Medicine. That has an impact factor of 70.67. The objective of the whole entire trial was to evaluate the efficacy and safety of the drug Velexator as compared with placebo in adults and adolescents with sickle cell disease. So the funding was by Global Blood Therapeutics. This is also the drug manufacturer for this drug. Some background on sickle cell disease and this drug in particular. About 70,000 to 100,000 Americans suffer from sickle cell disease. This is also highlighted in the trial. They talked about it in their background a little bit. It's a really hard to treat and aggressive disease, and there's not a lot of treatments on the market currently. Most people are either taking hydroxyurea or they're on this new drug that was pretty recently FDA approved in 2017. It's called Indari. It is L-glutamine. So there are 4 studies that have been recently published or are still ongoing that have to do with sickle cell disease. One is this one. There is an alternate HOPE study that specifically targets kids. And that one, I think it was published around the same time as this one. It went on in the same period of time. And then there's one for another drug that's about to be FDA approved. It's called the SUSTAIN study. So it's a drug that's pretty similar in mechanism of action to this one, but just a little bit different. And then there's also a study that's ongoing for a cure for sickle cell disease. So those are the current background studies around this. So sickle cell stems from the production of defective sickle hemoglobin, which is abbreviated HBS. The hemoglobin polymerizes when deoxygenated, and this results in overall damage to the red blood cell. Velexator is an HBS polymerization inhibitor, so this reversibly binds to hemoglobin to stabilize it. So this is as far as methods go for this trial, it's a multi-center phase 3 double-blind randomized placebo-controlled trial. It's a 3-arm trial, so they used a 1,500 milligram amount of the drug in one patient population, and then a 900 milligram of the same drug in another patient population, and then the placebo. Conduct of the trial was overseen by a contract research organization. Because the drug manufacturer funded this, they decided to have a lot of outside people kind of make sure that everything was straight and narrow and that they did their job right and that all of their ethics were correct in the study. So the trial included a 28 to 35 day screening period, up to 72 weeks of treatment, and a post-treatment visit at around 4 weeks or a month. There are primary and secondary outcomes of the trial. The primary endpoint was the percentage of participants who had a hemoglobin response, obviously. They defined this as an increase of more than 1 gram per deciliter from baseline at week 24 in the intention to treat analysis. They had both intention to treat and per protocol analysis in the study. The secondary endpoints include the change in hemoglobin level from baseline to week 24, reticulocyte count and percentage of reticulocytes, and lactate dehydrogenase levels. And then also the annualized incidence rate of vaso-occlusive crisis. So there's obviously inclusion and exclusion criteria. You can refer to page 7 through 9 of the protocol for all the inclusion and exclusion criteria. Mainly, it's just they had participants that age ranged from 12 to 65 years old. All participants had a confirmed variation of sickle cell disease, a hemoglobin level between 5.5 and 10.5, and between 1 and 10 vaso-occlusive crises in the last 12 months. Those on a stable dose of hydroxyurea for the past 3 months prior to the trial also were eligible to be included in the trial, just because if that was their mainstay of treatment, then they wanted to keep them on that treatment during the duration of this. And then they kind of tested to see if this was a good adjunct for the hydroxyurea. Exclusion criteria was the participants who were receiving regular red cell transfusion therapy, had received a transfusion in the past 60 days, or had been hospitalized for vaso-occlusive crises within 14 days before providing consent. The efficacy assessment, hemoglobin occupancy, was defined as the percentage of hemoglobin bound by the drug. And then as far as any statistical analyses, alpha tests were used. There's 2 specific tests that I am going to try my best with the names of the tests. It's the Land-Demetz and the Cochrane-Mantel-Hansel test were used to calculate results as well. An alpha level was set at 0.0481, and that was used in the primary analysis. A hierarchical testing procedure was implemented until the p-value exceeded that amount. So they pretty much started with the percentage of participants who had a hemoglobin response and then kind of graduated to the absolute change in hemoglobin level, then the relative change in indirect bilirubin level, and the relative change in percentage and change of reticulocytes, and then lactate dehydrogenase levels. So if the p-values stay the same throughout those different kind of stages or things that they were looking at, then they would count that as statistically and clinically significant. So moving on to results, the enrollment occurred between January of 2017 and May of 2018. There was a total of 274 people enrolled at 60 institutions across 12 different countries. 90 of the people were assigned to the 150 or 1,500 milligram arm. The 92 of the participants were assigned to the 900 milligram arm, and then 92 were in the placebo group. Baseline characteristics are on table 1 of page 6 of the trial text. They seem to be pretty well balanced. And then as far as the efficacy of the treatment in the results section, for intention to treat, a hemoglobin response at week 24 was observed as a much greater percentage in the 1,500 milligram group. When looking at all of the confidence intervals in that, it seemed that everything was very statistically significant. And then there was a huge clinical significant response in that 1,500 milligram group overall in the whole trial. The adjusted mean change in hemoglobin level from baseline to week 24 was 1.1 gram per deciliter, which exceeded that original 1 that they were looking for. And in the per protocol analysis, a hemoglobin response was 59% in the 1,500 milligram group, 38% in the 900 milligram group, and 9% in the placebo group. So obviously a very much so a higher response in that 1,500 milligram group. The mean change in hemoglobin level was 1.3 grams per deciliter, and in that 1,500 group, and then 0.7 grams per deciliter in the 900, and 0% in the placebo group. So overall, also, acute anemic episodes occurred in fewer participants in each Velexator dose group than in those receiving placebo. And annualized incidence rates in the 1,500 milligram group were lowered by a factor of 3. Excuse me. adverse events. Adverse events not related to sickle cell disease that occurred or worsened in participants during the treatment occurred in 94% of the people in the 1,500 milligram group, 93 in the 900, 89 in the placebo. But a lot of these adverse events were very manageable. Like there was a lot of instances of headache and diarrhea, and so this can be alleviated with over-the-counter medication, etc. And not a lot of participants complained about this or dropped out of the study. There were 4 patients that had fatal adverse events, but these were later evaluated to not be due to the drug. And so there were not substantial differences in the percentage of patients who had sickle cell-related adverse effects among the 3 trial groups. So in conclusion, that 1,500 milligram dose did increase hemoglobin levels. It did reduce the incidence of worsening anemia as compared with placebo. It significantly reduced indirect bilirubin levels by week 24, reduced reticulocyte percentages, and hemolysis. So the incidence of vaso-occlusive crises did not differ significantly between the groups. So the weird or kind of interesting thing that I found about the vaso-occlusive crises is that so this drug had the ability to, normally the drugs that have been produced for this disease they worsen the vaso-occlusive crises because as I put here, it will, hold on. So the absence of an increase of incidence rate of vaso-occlusive crises despite significant hemoglobin level increases suggests that this group can raise levels without negatively affecting blood viscosity. And so it does normally affect that blood viscosity, and so that means that normally it will be thicker, and so you'll have more vaso-occlusive crises, but this one doesn't affect that, so therefore it's effective in that way. So strengths of the test or of the trial were that there were extensive tests and equations used to determine both the intention to treat and the per protocol groups. The setup of the trial being blinded and randomized, placebo-controlled, using 2 different doses of the drug and spanning across that many countries, obviously a strength. Limitations were baseline values were calculated and used as mean values for the data, both screening day and randomization day. Baseline values calculated at week 24 were used as the mean values for weeks 20 through 24. So if a piece of data was missing, then they would use the other data point found, which didn't seem fair. So there's probably some unreliable baseline results in there. But other than that, I didn't see a lot of other limitations. Personally, I think that the trial was very effective. I think they did it very well. The fact that they had those contract organizations where they would look at the trial and the ethics and made sure that everything was being done properly, even though the drug manufacturer did sponsor it and fund the entire trial. It is a very extensive trial, and it's been given a lot of good credit by physicians and etc. So hopefully this drug will be approved at some point. So that's all I have, and then I'm going to turn the camera on everyone so that they can ask their questions. Yeah, and I don't think I can. Okay.
Okay, so you mentioned that there is a kid study that happened as well. Do you know if there is positive results from that as well? Also, what is the range of the kids in the study as compared with the 12 to 65 age range of the study?
Okay, so the kids study is, there was positive results for that. They tested the same things, and so results were very similar, and the age range was from 4 years old to 17, and so there was a little bit of overlap because the age range for this one was started at 12, so that 12 to 17 group was kind of divided between the 2 studies, but it did show effective results. Anyone else?
So the patient that died, were they from the treatment group, or was it equal among both groups?
There was 4 patients that died, and I think that the trial, if I have it, it was one of them was placebo, and then one of them was in the 900 milligram group, and one of them, or the other 2, were in the 1500 milligram group, and so it was pretty equal. So that's why I didn't see it as an effect, they didn't put that in the results as it being drug related. Who else?
Oh, is it me? Okay. So you mentioned that the patients were able to use hydroxyurea in conjunction with the intervention. Were there other exceptions made, such as pain medications? They were allowed to use those medications?
Yes, they were allowed to use pain medications during it, if they were already on it and that pain medication was a part of their normal regimen. If they needed to be on something strong, obviously they couldn't monitor over-the-counter medications, but if these patients had prescriptions that were being used, then they could stay on them as long as they were proven successful in relieving their pain.
Thank you.
Okay. How exactly did they define hemoglobin occupancy percentage, and is there a formula?
Yes, there actually was a formula that they used. They put this into one of their tests that they also used for their statistical analysis. It was calculated by the concentration of the drug in red blood cells divided by the concentration of hemoglobin in red blood cells to see the percentage that was bound.
So my question is, why did you choose this trial over the SUSTAIN trial that you talked about in that new medication?
The SUSTAIN trial was something that was more recently published, but I liked this one because it had a kid study and an adult study, and both of them seemed to be very positive results, and I feel like they were more thorough about their study, so I enjoyed reading about it more and learning about it, so that's why I chose this one.
Very good.
Thank you so much.